My name is Nora Yang. I'm the CSO, Chief Scientific Officer of Sensorion, and I'm delighted to have this opportunity to tell you a little bit about our company. So here's the disclaimer. Sensorion is a clinical stage biotechnology company. We're currently listed on Euronext. We won IPO a few years ago. So we specialize in therapeutic solutions for inner ear disorders, and our goal is to build a hearing care franchise in which we provide innovative therapeutic approaches to treat patients who have lost hearing and prevent patients from losing hearing when we know that they're going to enter into situations that jeopardize their hearing. And also to restore hearing in patients who are born deaf or who have lost hearing permanently. So currently, we have a portfolio of a mixed modalities of therapeutics in our pipeline, including 3 AAV gene therapy products that I'll spend the next few minutes to give you an update. Sensorion has a fantastic management team. Collectively, we have a wealth of experiences managing therapeutic development in all modalities in large pharma companies as well as building small biotech companies. We have built internal preclinical platforms as well as internal CMC production capabilities to support our gene therapy products. So our gene therapy products in the pipeline, in our current pipeline, came out of the Hearing Institute at the Institut Pasteur in France from their 40-plus years research that in 2019, Sensorion and Institut Pasteur formed a relationship or alliance in which Sensorion has an exclusive right to develop technologies that come out of Institut Pasteur. And this, so Institut Pasteur is one of the world's leading institutions in deciphering the genetics and physiology of hearing in the past decades. There are 150 world-class scientists in the institute, and they are responsible for the first 2 genes that they have identified to cause hearing loss. And Professor Christine Petit, who heads the Hearing Institute at Pasteur, is chairing our scientific board of scientific advisor board as well. One of the unique features of Sensorion is our ability to have built extensive alliances along the value chain, literally from the bench to the bedside, to support therapeutic development for hearing loss. I talked about the Institut Pasteur on our translational research side. On the clinical research side, we have an alliance with Neckar Hospital, which is Europe's largest pediatric hospital, and it is also the EU reference center for monogenic forms of deafness. And we also have an alliance with the French Biomedical Research Institute, which has access to a military population at risk of noise-induced hearing loss. And through these alliances, we're running natural history studies for all forms of monogenic forms of hearing loss or deafness, and also looking for biomarkers for noise-induced hearing loss. So on the diagnosis and patient journey side, we've forged several relationships, and 2 worth mentioning. So we have an alliance with Cochlear, the global leader in cochlear implant. Together, we're developing a drug-device combination approach to treat patients who are getting cochlear implants and to preserve their residual hearing. And we just recently announced the alliance with Sonova, which is the world's largest retail chain for hearing aids. So together, we're running a natural history study for presbycusis, age-induced hearing loss. So just a quick review of the biology of hearing. The organ that's responsible for hearing is the inner ear, this tiny little blue organ embedded deeply inside our ears. And the inner ear is an enclosed organ, and the snail-looking like part of the inner ear is called cochlea, and cochlea is the organ that's responsible for hearing. Inside cochlear, there are sensory cells we call sensory hair cells, and they are responsible for translating the sound wave, the mechanical energy from the sound wave to electrical potentials that can be communicated through the auditory nerve into the CNS, so our brain can understand and make sense of the sounds that we hear. With the world population aging and also noise pollutions, the human population is losing hearing at an alarming rate. The World Health Organization recently published a set of data showing that 1.5 billion people worldwide are suffering from hearing loss currently, and that number is growing. By 2050, 2.5 billion people worldwide will suffer hearing loss. And that is a huge public health problem and a clear unmet medical needs. So why do we lose hearing, or how do we lose hearing? And here are some key facts about our inner ear or sensory health. Every one of us was born with a set number of sensory hair cells. We have about 12,000 outer hair cells that are responsible for amplifying the sound signal. And we have only 3,500 inner hair cells, and these are the cells that are responsible for transducing the sound wave energy into electrical CNS signals. And each of the 3,500 cells, inner hair cells, is tuned into a specific frequency. So together, they give us the richness of the sound that we hear in music and the ability to distinguish between voices and understand languages. And inner hair cells are terminally differentiated cells. Once we lose them, humans cannot regenerate inner hair cells naturally. So once we lose a specific inner hair cells, we lose that specific frequency and permanently and irreversibly. So how do we treat hearing loss? At Sensorion, we take a holistic approach looking at hearing not as a single disease, but as many diseases depending on the cause of how people lose hearing. And then we engineer specific customized therapies to treat, restore, or prevent hearing loss from patients who are at risk. So here is our current pipeline, and we have a small molecule in the clinic that treats and prevents hearing loss in patients caused by environmental factors. And in the restore pillar, that we have several gene therapy products in development, which I'll focus presenting some of the, at the top level, what we're doing and where we are for the purpose of this conference. So about half of the hearing losses are caused by genetic factors, and the other half caused by environmental factors. And there are no fewer than 150 loci on the chromosomes that and no fewer than 85 genes that have been identified to be causative for hearing loss. And to understand these genes and when they occur, when mutations occur, and how to replace them or correct them will be the driving, guiding our therapeutic development approaches. So currently we have 3 gene therapies using AAV replacement therapy approach because the technology is mature. There are approved AAV gene therapies on the market, and we know how to manipulate capsids, and we know how to engineer specificities. So the first gene therapy product we have under development is targeting the Otoff gene. Otoff gene encodes a protein called otofurlin, and otofurlin is a calcium sensing protein in the inner ear, in the inner hair cells. And the otofurlin protein is a multi-C2 domain protein, and that little chain structure you see on the left, and it senses calcium levels. When calcium level is low, it's in the closed form, and it cannot interact with other proteins. But as the calcium level increases, otofurlin opens up, and it engages proteins, and particularly the synaptic vesicle, and brings it to the presynaptic membrane and causes fusion and the release of neurotransmitters. And the neurotransmitters will send the signals through the auditory nerve system and into the brain. So clearly, otofurlin plays an important role in this signal transduction process for hearing. And as you can imagine, when something goes wrong with otofurlin, and patients lose hearing. So the question is then, can we restore otofurlin in otofurlin deficiency patients, and can we restore functions? And here is one of the experiments that we've shown proof of concept in mice. So we use otofurlin knockout mice, and when you look at otofurlin expression in inner hair cells on the left panel, and you see the wild-type animals have abundant otofurlin expression in every single hair cells. And in otofurlin knockout mice, in the middle panel, and you see completely lack of expression of otofurlin. When we treated those mice with AAV-delivered otofurlin gene, majority of the inner hair cells regained expression of otofurlin, and that re-expression of otofurlin leads to functional recovery of hearing, which is demonstrated in the lower left panel of ABR measurements. And again, on the left, you see the wild-type mice that have this very regular pattern of waves that at all frequencies, indicating that they're healthy, they have healthy hearing. And the otofurlin knockout mice in the middle, for every single frequency, and the way it's flat, so they're completely deaf. And the AAV-treated, otofurlin-treated animals on the right recovered their hearing patterns. So reinstitution of the protein, of the otofurlin protein, can rescue hearing, at least in mice. And so we are eager to take that into patients, and hopefully this will provide much needed therapy for patients who were born deaf and suffer from this really devastating disability. The second gene that we're developing or we're correcting is the Gjb2 gene. And Gjb2 gene encodes a protein called connexin 26. And connexin forms the gap junction, and which is important or critical for tissue homeostasis in the inner ear. And it's also responsible for potassium recycling in cochlear, and again is critical for the sound signal transduction, plays a key role in that process. Gjb2 gene has attracted a lot of attention in companies because it's the single largest deaf gene that is responsible for half of the congenital or half of the hereditary deafness in patients. So research coming out of Institut Pasteur has really shown some breakthrough observations that basically they found that some deaf genes, including Gjb2, a single gene can cause a continuum of clinical manifestation depending on the type of mutation of the gene. So on one hand, on one side, for autosomal recessive mutations, you will see the congenital severe hearing loss. On the other hand, in heterozygous dominant autosomal dominant expression patterns of Gjb2, you will see the earlier onset of presbycusis. Those are patients who lose hearing in their 30s and 40s. So with a single gene that we have different clinical populations that we could use gene replacement therapy to treat. So we're currently devising products that target each of the population with precision, and we're seeing some very promising results that we hope to provide therapeutic relief for patients who suffer hearing loss because of this particular gene. So that is the end of my presentation. I don't think I have any time for questions, but if you have any questions, I'll be happy to talk to you for the rest of the conference.